Welcome to the EvoEd COVID-19 video series. In this video, we'll take a look at the anatomy of the SARS-CoV-2 virus. Then we'll look at the life cycle of the virus, from entering the cell to replicating and then to leaving the cell and eventually spreading to new hosts. By the end of this video, you should be able to list and describe the three major external and internal anatomical components of the SARS-CoV-2 virus. And you should also be able to explain the life cycle of SARS-CoV-2 by describing the processes of attachment, membrane fusion, replication, and finally shedding. Let's start off by looking at the outside of a SARS-CoV-2 virus. The virus is surrounded by a viral envelope, which has the main function of protecting the virus. It's made of lipids similar to a living cell membrane. It also serves the function of a platform for the major structural proteins to bind to, such as the spike proteins, which are used to attach to the receptors of hosts and to break in the host cell membrane. It's also surrounded by envelope proteins, which are there to help assemble new viruses from the proteins that are eventually created within the living host cell. Inside the virus, we have the RNA, which contains the genome or the genetic material of the virus, and it's stored in a winding continuous strand. We also have the nucleoproteins, which aid in RNA transcription and replication once the RNA is placed into the host cell. Before we take a look at the life cycle of a virus, let's look at a few of the cellular components that are involved in viral replication. First, we have the cell membrane. The cell membrane is a lipid bilayer that protects the cell from external molecules such as viruses. And within the membrane, we have embedded proteins, which can interact with molecules outside of the cell, such as viruses, which use these proteins to attach to the surface of the cell. Next, we have polymerase, which is an enzyme that uses a template strand to construct new RNA or DNA molecules, and the virus will use polymerase to replicate its own RNA. Next, we have the ribosome, which translates RNA into an amino acid chain and eventually a full protein. And the virus will utilize ribosomes to translate its own mRNA into proteins. And finally, we have the Golgi apparatus. Now, typically, the Golgi packages proteins in a lipid bilayer before it's excreted by the cell, but viruses will utilize the Golgi apparatus to export themselves from the cell. The life cycle of the cell begins when a new host is infected and the virus is able to attach to the surface of one of its cells. Spike proteins attach to receptor proteins on the cell membrane of the host cell. SARS-CoV-2 specifically targets ACE2 receptors, which are especially abundant on cells in the respiratory tract. Once the virus is attached to the receptor, a membrane enzyme, TMPRSS2, cleaves the spike protein, which allows it to begin to destroy the cell membrane. The cleaved proteins destroy part of the host cell membrane, providing access to the inside of the cell. Then, they facilitate the fusion between the lipid bilayer of the cell membrane and the viral envelope. Once the membranes are fused, the viral RNA and the nucleoproteins enter the host cell. Once the genomic RNA of the virus is in the host cell, it can then begin the process of replication by using the host cellular machinery. Before the virus can begin to replicate, it must first create polymerase for itself. Positive sense RNA from the virus uses the host cell's ribosomes to create virus polymerase enzymes. Next, the virus needs to make mRNA in order to produce new proteins. Since mRNA is positive sense, it can only be transcribed using negative sense RNA. Therefore, the virus transcribes negative sense RNA from its positive sense RNA using the new virus polymerase. This process is aided by the viral nucleoproteins. Now, the negative sense RNA is used to create several subgenomic mRNA strands that code for the specific proteins of SARS-CoV-2. Subgenomic just means that individual genes are being transcribed into mRNA sequences for specific proteins. The negative sense RNA is also used to create complete genomic positive sense RNA strands, which will be used as the genetic material for the newly replicated viruses. Just like DNA replication in living organisms, heritable mutations may occur during this stage. The produced viral mRNA strands then use the host cell's ribosomes to create all of the proteins necessary to build a new virus. All of these new viral proteins are then assembled with the help of envelope proteins in the host's rough endoplasmic reticulum, and viral replication is complete. Once the process of replication is complete, there's only one thing left to do: leave the cell and infect new hosts. The host cell's Golgi bodies package the virus into vesicles and escort the newly completed SARS-CoV-2 virus out of the host cell's membrane. So what happens to a virus after it leaves the cell? After leaving the cell, the newly formed SARS-CoV-2 virus can meet four potential fates. First, if it stays in the host body, it may be destroyed by the immune system of the host, or it might infect new cells within the same individual. If the virus is exhaled in a water droplet into the air, then it may be inhaled by a new individual and infect cells in a new host, or if it's not inhaled, it will eventually just degrade in the environment. So that's the life cycle of SARS-CoV-2. In the next series of videos, we'll take a look at the genome of the virus and discuss how mutations can lead to the evolution of new variants. Well, that's all for this one, and we'll see you in the next video.